We're going to try to finish up chapter 15 today, you guys. So I might go a little quick, but want to get this one done. When we finish the notes, we were talking about when the chromosomes don't separate properly. Okay, that's called non-disjunction. And if one that's missing one of the copies, in other words, it doesn't have 23 like it should, it only has 22. If it gets fertilized, it's called a monosomatic zygote. It's missing one. Okay, if the one that has the extra one gets fertilized, it's called a trisomic. Okay, because what's going to happen is it's going to have 3 copies of a chromosome instead of 2, or in this case, it's only going to have 1 copy of a chromosome. All right. So, and there's disorders that we're going to talk about that relate to that. Okay. Polyploidy, again, is anytime you have more than 2 complete sets of chromosomes. Okay. It's not, super common, but it does happen more than you think. Okay. So triploidy, 3N instead of 2N. That's what we're supposed to have. You're supposed to have 46 full chromosomes. But if you have 47, you're triploidy. And usually they say the chromosome. Okay. If it's, the second one or the 5th one, usually you guys, it's not 1, 2, 3, 1 through 6, because those are really big chromosomes. And if you get an extra one of those or you're missing one of those, that's a miscarriage. Okay. But it can happen in like trisomy 21, you've probably heard of Down syndrome. It usually happens in the smaller chromosomes if it happens. Okay. Tetraploidy, that could happen in plants, but it won't happen in animals. Okay. So again, it's common in plants, but usually if something like that happens in animals, let's say instead of getting 46 chromosomes, you have another 23. So you have 69 chromosomes. Not going to develop. Okay. That would be triploidy. Tetraploidy, it would mean you have 94 chromosomes. And again, that can happen in plants, but it's not going to happen in animals. Okay. It's weird that extra is more normal than missing. Okay. So there's more cases of trisomy, an extra chromosome developing, than there is when they're missing one, when they only have 45. The only place where that's more common is in the sex chromosomes. Okay. All right. So what happens? Well, sometimes just the chromosome itself. Now, when we're talking about non-disjunction, we're talking about whole chromosomes missing, but we can be missing pieces of chromosomes as well. Okay. There is a deletion where part of a chromosome segment is removed. Okay. And it depends on what that piece is responsible for, what kind of characteristics that individual has. Okay. There's duplication where they actually duplicate it so they have a little extra piece on there. Okay. There's inversion and I have diagrams of these, okay, so I'm going through them kind of quick and I'll show them to you more in a minute. But inversion reverts the original orientation, so it flips it. Okay. And then there's translocation, where a piece breaks off from one and goes to another. Now, if you get that one with that extra piece on it, there's going to be a problem. If you get the one that's missing a piece and you end up getting the one that has the extra piece, then you'll be fine. They're just in different places, but their chances of that are actually pretty slim. Okay, so here, deletion. Excuse me. During meiosis, a piece is missing. Okay, and there's a lot of disorders with this. My youngest daughter has a deletion problem. She has something called Prader-Willi, where she's missing part of her 15 chromosome. Okay, so A, B, C, E, we're missing D. Okay, and then you can see the chromosome is shorter because it's missing a piece. Okay, duplication, look at A, B, C, D, E, A, B, C, B, C. So this one's going to be longer because it's got extra information in there. Okay, and again, when the RNA comes in and it's supposed to make a copy of the D and then it goes to the spot it's supposed to be and B is there's going to be a problem. Okay, inversion. You can see here, instead of A, B, C, D, it's A, D, C, B. This part got switched around. It usually doesn't happen to a whole chromosome. It's usually a part of it. Okay. And again, this will throw off when it comes to make a copy. If it's coming to make a copy of B and D is there's going to be a problem. And then there's translocation. See where this piece, okay, you have your 2 chromosomes here, But what happens is AB attaches over here and this attaches over here. They switch spots. Okay, so a piece of this ends up, you know, it's supposed to be A, B, C, D, E. This is the order of the genes, and it ends up A, B, P, Q, R. You know, it's in the wrong order. It's in the wrong place. Okay, and again, if these get inherited, then there's going to be some issues because of it. Okay, so some of the human disorders that are caused because of chromosome alterations. Again, you need 46 full chromosomes. If you're missing a piece, if you have an extra piece, if you're missing a whole chromosome, if you've got an extra chromosome, there's gonna be some issues, okay? Most issues are so severe that the child is miscarried, okay? But there are many cases where children are born with these chromosome abnormalities, okay? Some of the aneuploidy, in other words, they're missing, with the non-disjunction, it's really gonna mess up a lot of systems. It messes up a whole bunch of stuff, not just one little characteristic, okay? Now, when we're talking about characteristics like if somebody is an albino, that's not a chromosome thing, you guys. That's a gene thing, okay? They inherited a recessive gene. But when we're talking chromosome things, there's all kinds of characteristics that get affected because of it, okay? So it's not just one or two things, it's many, okay? If the individual survives with this chromosome issue, there's many of them that have already been named and characterized and whatever, but they have some sort of syndrome. And I think I have a picture. The most common, you guys, is Down syndrome. And I know you guys know what Down syndrome is. What happens with Down syndrome, they get an extra 21 chromosome. It's called trisomy 21. Okay, and that's a small chromosome. 21 is a small chromosome. So they get an extra one. And again, you think, well, what's the big deal? Extra chromosome can't be bad. It's very bad. Well, not really. Down syndrome people are wonderful people. They're the most beautiful people you'll ever meet. But they have a lot of issues. Many of them are mentally challenged. A lot of them have heart conditions. They have characteristics like their tongue is larger than their mouth. So it always looks, they used to call them mongoloids, which is awful because they had that characteristic face. But basically, a lot of times you'll see their tongue out because it's too big for their mouth. They have extra eye folds, on their eyelids. There's characteristics because of having that extra chromosome, okay? And you can see 1 out of every 700 children born in the United States has Down syndrome. It's not as uncommon as you think. Like I said, my daughter has Prader-Willi, which is a deletion of her 15th chromosome. She has just a little piece she's missing. And I'd never heard of it when she was born, but it turns out 1 in 10,000. It's not as uncommon as I thought it was, okay? So there's all kinds of characteristics because of if you're missing chromosome pieces, if you have extra chromosome, okay? The frequency of Down syndrome usually increases. See, non-disjunction usually increases as the mother gets older, okay? So when the meiosis occurs, as you get older, it's not quite as efficient, and that's when you end up with chromosomes not separating the way they are. I think it's 35 and over, they say, you have a more chance of having genetic disorders. Because remember, your eggs, when you're female, were there when you were born. Okay, so they're getting older, and so there's going to be more issues with those eggs. Okay, and there's a beautiful Down syndrome child, and you can see here's the karyotype right here. They have an extra chromosome. Okay, and again, they have the characteristic eyes, their mouths and stuff, but oh, they're beautiful people. Anyway, that's one example of a trisomy where they have an extra chromosome, and that's the most common, okay? With the monosomy, if you're missing a chromosome, the most common place that occurs is in the sex chromosomes, okay? Sometimes females are XXX, and it's really weird. They used to call them super females because they have an extra X chromosome. They tend to be pretty healthy. The biggest characteristic I've read that they have is sometimes they can't have kids, okay? There's Klinefelters, which is, if you see, they get XXY, so they don't separate. They have an extra chromosome. They have 2 X's and a Y. This individual is a male because they have the Y chromosome, okay? And these guys have a lot of characteristics. Usually they're infertile. They're usually smaller. They have some mental challenges. There's all, and again, extra chromosome, wouldn't think that would happen, okay? With women, there's something called monosomy X. They just get an X. They don't get an XX. They don't get an X. They just get 1 X. And it's called Turner syndrome. And a lot of times they'll write it as XO because you're missing a chromosome. Most women with Turner syndrome are smaller. They don't really develop very well sexually. They don't get a lot of their characteristics. They're sterile. They can't have babies. Okay, so there's issues because of it. Okay, so those are some. And again, with the monosomy, that usually only occurs in the sex chromosomes. It's not very common in the autosomes, in the normal body chromosomes. Okay, with altered, cry du chat is the cry of a cat. They're missing a piece of their 5th chromosome. They have like cleft palate, cleft lip. They have usually a hernia into their diaphragm. There's lots of issues, and a lot of these guys, if they make it through birth, don't live very long. Okay, they're severely disabled, and again, they have a cat-like cry. That's where the name came from, the cry of the cat. But they usually die, if not soon after they're born, in early childhood. They don't live very long. Okay, there's some cancers that have been linked to translocations. Okay, now remember translocation, where a piece breaks off and attaches somewhere else. Okay, there's one of them is leukemia, chronic, I don't even know how to say this, myelogenous leukemia. Okay, there's certain cancers with the translocations. There's all kinds of disorders, you guys. I'm just giving you a few. Okay, so and this one happens is a 22 and a 9. Okay, what happens is a piece of the 22 breaks off and sticks on the 9. So you've got these two new, like almost like crossing over. But if you inherit this chromosome, you get extra. If you inherit this chromosome, you're missing some of this 22 because it attached over here. And if you don't get both of them, now if you inherit both of them, you're okay because you've got all the genetic information. It's just kind of in a goofy order. But if you get this one and not this one, or this one and not that one, you're going to have problems. Okay? So some inherited parents, and again, Mendelevian inheritance it's pretty straightforward. Okay? There's normal exceptions. Okay? One exception is genes that are located in the nucleus. Okay? That most of the genes that we talk about, once it opens, the genes are located outside of the nucleus, but sometimes you have nuclear genes. And in that case, the sex of the parent contributing the allele is the factor. In other words, and you'll see this in mitochondria too. There's mitochondrial DNA. If they test you for your mitochondrial DNA, it's your mother's. There's no father contribution. Because remember, mitochondrial alleles came from you when you were in your mother's womb. So you got her mitochondria. So they can actually use mitochondrial to trace mothers back from way back. So there are some different inheritance patterns. And again, that's in the organelles and in the cell itself. Okay. We also have what they call imprinting. There's a few traits where the phenotype passed on from the parents is located in the trait. An example of this is, like I said with my daughter, she's missing a piece of her chromosome. And it matters what chromosome, if it's the maternal or paternal, because we found another genetic disorder where it's on the same in chromosome, but it's a different characteristic depending on if the chromosome comes from mom or dad. So there's imprinting going on. Okay, there's various phenotypes, and it for some reason it has to be from those chromosomes. Okay, genetic imprinting involves silencing certain parts of the genes and using other parts of the genes. There's still a lot more research going on with this, and this one's kind of, a little bit iffy. But when we're talking about imprinting, genomic, when when a baby's born, they know that they're supposed to suckle right away, or you see animals, cows, horses, they get up right away, things like that. Those are all imprinted. Okay, and here's some examples again with mice. We're almost done, where genes get shut off and turned on. Okay, here, this is a bad gene, and this is a good gene, and this one is a normal sized mouse. But if the bad gene gets turned on, then you get a little tiny mouse, a little dwarf mouse. And there's all kinds of cases of that. Again, it's they look the same, but for some reason, one gene turns off the other one. And if it turns it off, then you're going to get a little dwarf mouse. Okay, so I'm getting there. Okay, so extranuclear genes or cytoplasmic genes are found in the organism, and this is when I was telling you about mitochondria. Mitochondria, chloroplasts, they have their own DNA. Okay, and sometimes there's extra genes in there, but you inherit those just from Ma. because that's where you're developed. And I hope, oh, man, okay. I'm not going to go that fast. I'm done.